All right, everybody, it looks like we are going to get started with our next session at 1 o'clock promptly. Hi, my name is Rachel Gravon, and I'm going to be moderating our afternoon sessions today with the help of Darren Brandt, who is working invisibly in the background as my silent moderator. So I hope everyone enjoyed their lunch. We're going to get started with our next session, which is one very near and dear to my heart. It is about algae. All right, everybody, it looks like we are going to get started. So let's get started. Our first talk is going to be with, I'm sorry, I'm getting a ton of feedback on my computer. Okay, there we go. I don't want to hear my own voice. That's weird. We're going to get started with Theo Dreher's talk. Thanks, Theo.
Thanks, Rachel. Thanks, everybody, for tuning in. Have we got the first slide up? Yeah, right. So this is work that we've done over the last few years. It's been really exciting for me. I retired at the end of, as a professor at Oregon State University at the end of December last year. And so as things were winding down in the last year, I was doing quite a bit of this work myself. And since I've been, during the pandemic, I've been working about half-time on this after retiring. But I have got a lot of help, and that's reflected here with Ryan Mueller's name. He's a colleague in the Department of Microbiology, and Ed Davis, who is in the Center for Genome Research by Computing at Oregon State University. So we've analysed cyanobacterial blooms, next slide please, the filamentous blooms of the Nostocalydes. So there's no microcystis in this work. And this slide shows the lakes that we've sampled. So it's mostly been Oregon. In the past, we've sampled Anderson Lake up there at the top left and determined the genome sequence of the Dolichospermum that produces anatoxin. That's Anderson Lake up near Port Townsend. And Browns Lake in eastern Washington was the one Washington Lake. The others, as you can see, are in Oregon, and there's one at the bottom, the Iron Gate Reservoir in northern California. And in Oregon, these are mostly alpine lakes or reservoirs on the major rivers draining the Cascades. So a fair number of lakes. And in the next slide, this outlines the procedure that we've used to determine the genome sequences. Our intent was not to get partial genome sequences, but complete so-called closed or finished genome sequences. So you get as much information about the genome as you can. So we've sampled, we've not cultured any of these strains. That's the usual sort of case. So we've done direct sequencing because it does have some advantages. It gives you, as we'll see, it gives you direct information on the standing population. And all of this has been done over the last few years. So these are quite recent HABs from the Pacific Northwest. So we've always documented microscopically what's been there. And then we've filtered the samples to get rid of small stuff, particularly free living bacteria. And then we've done two types of sequencing that's illustrated by, if you're bakers, by the decorative, we call them hundreds and thousands in Australia. So at the bottom there is the bigger ones. This represents long read sequencing. So the technology we've used there gets rather large segments. The idea being that genomes have repeat sequences that are maybe 4,000 or 5,000 nucleotides long, and these can confuse most the traditional sort of sequencing acquisition pipeline. And by getting these large chunks all at once, you maximize the ability to put them together again to produce a genome, because you have to physically fragment the DNA in order to sequence it, and then use the computer again to put together that fragmented information to assemble a genome. At the same time, we've done short read sequencing, and that's illustrated on the far right, where you get little snippets. You get tons of them though, whereas the long read sequencing, you don't get quite so much information. And the combination of the two gives you the sort of information, the rich information that I'll talk about, and we'll see what that can do in the next slide. So this is the, we'll go through these steps one by one. The questions that we were interested in is first, what cyanohabs, which of the cyanohabs do produce toxins or taste and odor compounds, and which ones? And you can infer this from the genes that you find. The second question, how does the morphotype, that is what you see in the microscope and describe as, say, an Aphanizomenon flossaculae or a Dolichospermum, how does that relate to the genotype? Because that's not obvious. It might be that you see one sort of morphotype and underlying that could be actually a few different strains, different genotypes. The third question, what's the cyanohab population structure in a given bloom? I just mentioned that. Could there be perhaps a number of strains? And what happens from year to year? Overwintering, the bloom virtually disappears and then comes back to a raging presence that fills the lake. So you go from boom to bust. And what happens there in a lake from year to year? The fourth question, how do cyanohabs differ from lake to lake across a region? If you're looking at say, Dolichospermum that has fairly similar sort of morphology under the microscope, do you find the same strains or are they fairly different? The fifth question, what's the taxonomic relationship between Anodina, Dolichospermum, and Aphanizomenon flossaculae? And this is a very thorny question that I'll get to at the end. The last question there is, what sort of co-occurring bacteria exist? There's the idea that there's a phycosphere, so that there are other bacteria and probably have a shared lifestyle with the cyanobacteria and perhaps provide some sort of benefits, maybe produce vitamins or something. And so there's a lot of interest in the field about this. Now, this I just won't have time to touch on. Next slide, please. I also won't discuss this one for very long, but the idea here is that if you look at that green column, there are a lot of entries. So we've really got quite a lot of genomes. And the ones that are dark green, they're the ones that we've completely finished. So those are completely closed and finished genomes. So that really all the information that you can glean from this type of an approach is available. In other times, say the lighter green ones, they're not quite finished. So the true arrangement of the genes around the chromosome is in a bit uncertain. I'll get to the details of this later on. Next slide. This shows these associated bacteria. And again, just the same thing. I want to say that we have quite a lot of these, and I won't be able to talk about that because of time limitation. So if we go to the next slide. Next slide, that one, yep. This is the first question. So what do we find in interpreting the genes present once we've finished these genomes in terms of the presence of toxin genes and taste and odor compounds? Because these are the compounds that are important to us in terms of getting good, clean drinking water and with respect to public health. So the good news, and you kind of know this, is that not all of these cyanobacteria are toxic. And perhaps certainly more than half of them, half of the isolates we've got are not toxic. We didn't find any toxin genes associated with Aphanizomenon flossaculae, and we have a number of those isolates, nor from Globiatrichia. Globiatrichia on the east coast is toxic, but perhaps not here, certainly not, we haven't found that to be. Amongst Dolichospermum, or I've got them entered here as Anodina, but think Dolichospermum, because these are all planktonic, these are all free-floating blooms. A number of these are not toxic, but some also are. And that includes anatoxin producers. Now, in this case, it's a Finnish isolate, but I mentioned previously we'd sequenced the genome from the Dolichospermum in Anderson Lake up near Port Townsend, and that's a potent anatoxin producer. We've seen cylindrospermopsin genes and microcystin genes. So all of those do exist in the Pacific Northwest, just not in all of the genomes. Geosmin is also present, but again selectively. And so we have 4 strains of about over a dozen that are geosmin producers. So some of the cyanobacteria, bottom line, is produce toxins or geosmin and have the genes to do so. Others don't, and they won't be able to produce any of those compounds. They just don't have the genetic capacity. Next slide, please. This shows geographically in Oregon where we've seen the toxin producers. So C for cylindrospermopsin, that was found in the Memorial Day bloom. It occurs really around Memorial Day every year in Detroit Lake, that's upstream on the Santiam River, upstream from Salem. And that, you might remember a couple of summers ago, there was a problem with drinking water in Salem. There was an emergency and there was a do not drink order for pregnant women and young infants. So there was low concentrations of cylindrospermopsin and microcystis that broke through into the finished water. So we've isolated here this genome sequence of the cylindrospermopsin producer and the microcystin producer. We also saw microcystin in 3 other lakes. 2 of those were the same. I'll talk about that later, the same strain in lakes that are a couple of 100 kilometres apart. And then on the bottom right, down there in the southeast on the map, that M relates to the picture of the dead cow over there on the left. This was a Dolichospermum producing microcystin that was associated with a steer die-off of about 30 steers in summer, I think it was 2018. So we see microcystin producers amongst Dolichospermum are sort of scattered in Oregon. There are a number of different strains. And the Globiatrichia producing isolates are shown. There were 2 Dolichospermum and 2 geosmin that were geographically distributed. Next slide, please. Now, the second question is how does the genotype, how does the genome sequence relate to morphotype with a microscope? And of course, we all know, if you've looked at these cyanobacteria, they're really beautiful under the microscope. They're easy to see because they're big cells and they make these great big long filaments and aggregations and colonies. And even with that crummy little cheap microscope there on the bottom right, which we take on all our field trips, it's fantastic. You can see all this stuff. So it's tremendously important to use morphology to see what you've got. And fortunately, what we've seen is the relationship, at least in these HABs that we've sampled, between genotype and morphotype is quite simple. Where there's a single genotype, morphotype, we found a single genotype. And in the case on the right, where there are 2, we've found 2. Now, what I would say you cannot do, though, is infer the genotype from the morphotype across lakes, because very similar looking morphotypes can yield very different genotypes. Next slide. The question of succession from year to year. What this shows is the genomes aligned from left to right. And then if the same color bar appears, that means the same sequence is present, the same genes are present in the same arrangement. And what you see here on the left for Detroit Reservoir, where we've sampled 4 years running, and twice in 1 year, we get the same strain every year. Very interesting. So overwintering, where you've hardly got any cells, and there could easily be a genetic bottleneck, and then it roars back to a new HAB, same strain. And that was also seen in Odell Lake over 2 years. So I don't know that happens all the time. It's probably not going to be totally generalizable, but it perhaps is more the default state, which is kind of nice because it means that if you get to know the HABs in your system, in your lake, that you could expect reasonably a certain amount of consistency. Next slide. On the other hand, if you compare the genomes of Dolichospermum on the top there, the genome diagram at the top, from different lakes, you get this jumble. So rather than one long pink bar, you get a whole lot of different colored bars, reflecting that the genome has been segmented. Each of those colored bars is found in each genome, but they're much smaller, shorter sequences. And if you then look at the lines that connect the 2 vertically, some of those are quite slanted, meaning that those gene segments have shifted from one part of the chromosome to a very completely different part. So that's the usual case for Anabaena. And that's why I say that the morphotype does not relate very well to genotype if you're sampling different lakes. Globiatrichia, on the other hand, doesn't seem to have quite so much genome fragmentation and randomization. Next slide. Occasionally, when you look geographically, you will find the same strain, evidently, because that's happened here, where we see the same strain as a microcystin producer from Odell Lake, and then from much further north in Lake Billy Chinook. Next slide. And so I'll just finish with this in a couple of minutes. When we look at relationships between genomes, there are two ways you can do it. You can build a phylogenetic tree that's like a family tree on the left, and this follows from inferring the sequence changes that accumulate between different genomes, and you infer the time-specific order in which they were incorporated, and you get a family tree. And what you see there, as we recognised in that paper in 2018, is that there's an ADA clade. That's A for Anabina, D for Dolichospermum, A for Aphanizomenon floss aquae, that is that coloured branch that represents most of our genome sequences from our standing halves. On the right reflects the analysis of average nucleotide identity. And there you take these aligned sequences and you see how many changes have occurred. You quantitate the changes. And then you compare them on the graph, the list of genomes vertically and horizontally. And the diagonal represents the self against self. And then there are relationships where you get boxes. And those boxes, the largest box there that's mostly fairly yellow, that reflects that ADA clade. And you can see that those boxes are very separate from other genus or species boxes. So in the next slide, what you the left branch reflects just reproduces what we had on the previous slide. On the right branch is a rather large that was published earlier this year from a group in Finland. I'm not presenting our new tree that incorporates all these new sequences because we're just about to submit that, but that's reflected in the middle. The bottom line is that all of these Aphanizomenon floss aquae and Dolichospermum blooms we're finding are from the same branch of this Nostocales family of cyanobacteria. So they're all fairly closely related. And the evidence is that this branch ought to be considered a genus. A genus normally has one name. It doesn't have an Anabina name, a Dolichospermum name, and an Aphanizomenon name. So there's a taxonomic problem. And further, we've found that Cuspidothrix, Isachin koi, is also included in this. So there are some problems that we can identify from sequencing that were introduced into taxonomy over the previous period. And we think there are probably 10 different species within this genus. We can talk more about that in questions. The next slide just is a summary. I don't think we need to go over that. And then finally, if you go to the last slide, yes, the next slide, the acknowledgements. I mentioned my colleagues Ryan and Ed Davis. There have been a few other people, and particularly a lot of people helped with providing samples. In Washington, Sally has, and Fran Sweeney from, we've had a long relationship, long interactions at the King County labs. So that's about it. Thank you.
Thank you so much, Theo. That was fantastic. I did forget to mention earlier that we will be taking questions at the end of all of our talks, as we did for the previous sessions this morning. So up next, we have a recorded presentation from Angelica Lucchetto. And Angelica is here to answer your questions after the presentation as well. So take it away. Thanks.
Hello everyone, my name is Angelica Lucchetto and I am an undergraduate student in environmental science at the University of Washington, Bothell. Today I'll be presenting the influence of floating logs on lake ecosystems, a comparison of Coldwater Lake and Spirit Lake on Mount St. Helens. And I've been a part of this research project for 3 years now, so I'm excited to present our findings so far. I'd like to thank the faculty members who've helped me with this project, which are Avery Shinneman, Kenneth Fox-Dobbs, and Jim Gawel. All Right, as a little bit of an introduction, Mount St. Helens erupted on May 18th, 1980, and it eliminated almost all vegetation in the blast zone. The debris avalanche and pyroclastic flow, which you can see in beige and pink in the top picture, altered the blast zone's hydrology. The pyroclastic flow blocked off the outlet of Spirit Lake, which raised the surface elevation by 64 meters. And also the debris avalanche blocked off Coldwater Creek, which formed Coldwater Lake itself. Although Coldwater wasn't hit directly by the pyroclastic flow, the lakes are comparable because their most recent geologic configurations were formed from the same event. The key feature that distinguishes the lakes is the presence of the floating log mat on Spirit Lake, which is outlined in red on the bottom left picture. Our original research focused only on Spirit Lake, but we decided to compare them because Spirit Lake was inaccessible this year due to COVID camping and travel restrictions. But it actually ended up being a good opportunity to compare otherwise similar lakes that have one intriguing difference. So the main question or goal of the study is, how does Spirit Lake's floating log mat impact the lake's ecosystem? So our original reason for sampling at Spirit Lake in the first place was Jim Gawel's previous work, which found that there was a nutrient imbalance where there was far more nitrogen output than nitrogen input, which was pretty unexpected. Our original goal was to determine the source of the excess nitrogen, and we had the hypothesis that the nitrogen-fixing cyanobacteria on the log mat could be a major contributor. We did some rough calculations based on our results from 2018, but we didn't end up finding cyanobacteria in a high enough abundance to prove that hypothesis from log mat growth alone. However, our sampling prompted continued research on the role of the log mat, which is the topic of this presentation. Sampling the log mat also gives us a better idea of the lake's ecology, as sampling has been pretty spotty between the 1980s and now, and it's only targeted plankton suspended in the water column. So by sampling the periphyton, we're learning more about the unique habitat niches in the lake. The methods we used to get a comprehensive sample of the lake's ecosystem included plankton tows, surface sediment samples, macrophyte samples, and log scrapes, and a suspended rack of microscope slides beneath a smaller log mat that we constructed. And we constructed the smaller log mats in Duck Bay out of logs that were already floating on the lake, which we roped together and secured to logs sticking vertically from the lake bottom, which you can see Jim doing on the left there. Because the constructed log mats were secured in Duck Bay, it would be easy for us to return to them and retrieve samples throughout the summer. So here are some of the most common taxa we saw in both plankton tow and periphyton samples from Spirit Lake. These taxa represent several algal groups, including diatoms, chrysophytes, green algae, and cyanobacteria. And these particular taxa were found in different proportions in plankton tow samples and periphyton samples. For example, nostoc, which is the primary type of cyanobacteria we found, was much more abundant in the periphyton because it has to grow attached to a substrate. From our 2 years of sampling at Spirit Lake, we noticed that taxa diversity in plankton tow samples was highly variable from year to year, as can be seen in the bottom two graphs. The top graph shows taxa richness on the log mat for 2018 and 2019, which was also highly variable from year to year. Unfortunately, since we only have 2 years of data, we can't draw any conclusive patterns about diversity or richness, though. However, we did notice that log mat periphyton samples displayed much more taxa diversity and richness than plankton tow samples. Cyanobacteria made up an average of about 30% relative abundance in periphyton samples, and were even present in plankton tow samples, although to a much lesser extent, which is represented by the tiny 2% red wedge of the July 2019 pie chart. The presence of cyanobacteria is actually unusual for an alpine oligotrophic lake, which is further evidence that the log mat provides unique ecosystem niches. And here are the sediment results from Spirit Lake. So the graph on the left shows the concentrations of carbon and nitrogen from dredge sampling at various locations around Spirit Lake. Although there's low organic content in the sediments overall, the highest concentrations of both carbon and nitrogen are found in the west arm, along the east shore, and in the case of nitrogen, in Duck Bay. Looking at the graph on the right, we can see that throughout the summer and into the fall, the log mat is most frequently found in the east and west arms of the lake. So since the highest concentrations of carbon and nitrogen were found in those areas, it's likely that the log mat is a major contributor to those nutrient inputs. All right, so now moving on to Coldwater Lake. Again, our decision to sample at Coldwater as a comparison to Spirit Lake really stemmed from the limited access to Spirit Lake this year due to COVID. But fortunately, we can use Coldwater Lake as a kind of natural control model to see how the presence of the log mat impacts Spirit Lake. So to do that, we use the same plankton tow surface sediment and macrophyte sampling techniques that we used previously. But to get periphyton samples from Coldwater, we scrape the periphyton off of partially submerged rocks that we collected from the shoreline. And here are the most common algae taxa we found in Coldwater Lake. Like Spirit Lake, the most common taxa all come from different algal groups, but the last three on the list differ between the lakes. However, all the algae taxa that we found in Coldwater were also found in Spirit Lake, so there weren't actually any taxa unique to Coldwater. Interestingly, as the pie charts at the bottom show, the plankton tows exhibited greater taxa diversity than the periphyton samples, which is the exact opposite of what we saw in Spirit Lake. Our Coldwater Lake samples also support the idea that Spirit Lake's log mat provides unique habitat niches, as we found no cyanobacteria in either the plankton tows or periphyton samples. Again, though, our sampling was extremely limited. We only sampled a small area of the lake and only got 2 days worth of samples, so the comparison between the lakes is imperfect. When comparing the two lakes to each other, we found that plankton tow samples are similar in terms of taxa richness and diversity. However, Spirit Lake periphyton samples displayed greater diversity of taxa and algal groups than Coldwater Lake samples, as shown in the graphs on the left. This could be because the log mat is a more ideal substrate that provides more habitat niches than the partially submerged shoreline rocks that we sampled from in Coldwater Lake. And again, cyanophyta were only present in Spirit Lake. So moving on to the sediment samples, the graph on the left shows the carbon to nitrogen ratio of the items we sampled from both lakes. It demonstrates that percent carbon and percent nitrogen of primary producers are pretty comparable between the lakes, as shown by the circled groupings. It also notably shows that both lakes have low organic content in the sediments, which is to be expected in alpine oligotrophic lakes. The graph on the right shows the range of percent carbon values found in each lake. As you can see, there's a larger range of percent carbon values in Spirit Lake, notably on the higher end of the range. This indicates that the log mat contributes to the higher nutrient levels in the lake. Overall, the sediment records show that the log mat does create spatial heterogeneity in the lake ecosystem. All right, so to wrap everything up, here are the key takeaways from the past 3 summers of research. Firstly, the taxa present in plankton tows from both lakes are pretty similar in richness and diversity. Spirit Lake has a slightly greater overall number of taxa present, but that's likely due to more comprehensive sampling from Spirit Lake. Next, taxa diversity in benthic periphyton differs greatly between the lakes, with the Spirit Lake log mat hosting a greater number of taxa and algal groups than the Coldwater Lake shore rock periphyton. Most notable is the presence of cyanobacteria in Spirit Lake, but not in Coldwater Lake. And finally, the floating log mat on Spirit Lake contributes to the heterogeneity of the lake's ecosystem by depositing organic matter from shedding periphyton and log debris in the areas the log mat occupies most frequently. All right, so here are the references that I use for this presentation. Thank you all so much for listening, and I'll be available on Zoom now to answer any questions you might have.
Great. Thank you so much, Angelica. And again, another reminder that we will be doing our Q&A at the end of the session, so please send your questions to info@walpa.org. Next up, we have another pre-recorded session with a speaker, Sarah Norberg, who will also be available afterwards for questions. So let's get started with that one.
Wapato Lake, located in South Tacoma, is a small urban lake that has been plagued with ongoing water quality problems since the late 1900s. The primary factor contributing to poor water quality, toxic algal blooms, and repeated closures of the lake from recreational use is the input of excessive nutrients, primarily phosphorus. This harmful nutrient, derived from urban sources such as lawn fertilizers and pet waste, enter the lake in stormwater runoff. Efforts have been made over the past 50 years to improve water quality in Wapato. Removal of aquatic plants, dredging, alum treatments, and even the introduction of drinking water have been mostly ineffective or temporary in preventing toxic algal blooms. In an attempt to limit intrusion of harmful nutrients from urban sources, a diversion dam was installed, creating two separate basins, north and south. The North Basin acts as part of Tacoma's stormwater detention and conveyance system. An estimated 390 million gallons of primarily stormwater runoff from a large urban area enters the North Basin each year, carrying with it approximately 150 kilograms of phosphorus. Around 50% of the phosphorus entering the North Basin exits through a bypass drain, where it then enters the city's stormwater system. That phosphorus-laden water then flows directly to Puget Sound.
Hi, my name is Sarah, and I'm an environmental specialist for the City of Tacoma in our stormwater group. Whether it's through stormwater sampling or public education and outreach, my job is to help prevent stormwater pollution. The influx of harmful nutrients into our stormwater runoff is an issue of growing concern. Changing human behavior to prevent the introduction of harmful nutrients into stormwater runoff is the ultimate solution. However, actions need to be taken now to protect affected water bodies like Wapato Lake, as well as other lakes, streams, and Puget Sound. Managed attached algae production has been used for over 50 years as an effective and low-cost means for filtering water. Algae turf scrubbers are very popular in the reef aquarium community as an effective biofilter, which is how I learned about them. One of my hobbies outside of work is building and maintaining reef aquariums. The concept behind an algae turf scrubber is simple. Water is pumped across a mat or screen at a controlled rate. With proper flow rate, exposure to light, and nutrients in the water column, algae growth is promoted. As the algae grows, it removes excess nutrients from the water and incorporates it into its tissues. Once the mat or screen has reached loading capacity, the algae is harvested, and the excess nutrients are permanently removed from the water. I started to wonder if an algae turf scrubber could be built that could remove excess nutrients out of an infected lake like Wapato. After searching through the literature, I found many cases of large-scale algal production being used to filter effluent from everything from wastewater treatment plants, dairy farms, and overland runoff. I approached the University of Washington, Tacoma with my idea, and much to my surprise, they said, let's give it a try. The objectives for our project were to design and build a small-scale floating algae turf scrubber for deployment in Wapato Lake, establish algae or native periphyton growth, and determine the uptake rate of total phosphorus through periphyton growth.
Our initial design for the scrubber included a 3 by 5 foot frame with 3 separate mat surfaces, all positioned at approximately a 2-degree angle. We mounted the frame on a dock float and attached a 6-inch drainage pipe intended for sample collection and flow monitoring. Each mat section had its own 500-gallon-per-hour pump and spray bar. The scrubber would be tethered to an anchored john boat in the North Basin. The john boat would hold the solar panels, batteries, and sampling equipment. We had hoped to deploy the scrubber during the peak growing season in July 2019. However, due to delayed funding, we were not able to deploy the scrubber until early October. Right away, we started having issues with power due to the shorter days, increased cloud cover, and rain. The single 100-watt solar panel was just not enough to keep the pumps flowing 24 hours a day. Another issue we ran into was maintaining a consistent 2-degree slope on a floating structure with water flowing over it. The drain pipe we added at the base of the scrubber retained the water long enough to shift the weight of the entire platform. Conversely, any wind or additional water from rain would cause the entire platform to shift back and divert the water flow over the back of the platform as opposed to across the mat surface. Through a lot of trial and error, we were able to fix the balance issues. However, even with an additional 200 watts of solar, we could not keep up with the power demands of the 3 pumps. We removed one pump and the mat in the center, but flows were still intermittent, especially at night. We were successful in establishing some periphyton growth, but the colony was primarily made up of diatoms, and our average dry weight of collected material was less than 2 grams. We took this first deployment as a good in-field trial run and decided to try again the following summer, when we could deploy earlier in June. For our next deployment, we removed the dividers from the scrubber to create one single mat surface that was approximately 1 meter square, fed by one 2,000 gallon per hour low amperage pump with an inline valve to adjust flow rate. We installed larger solar panels for a total of 400 watts and 3 110 amp hour batteries. The outlet drain pipe was also removed, which allowed water to flow off of the scrubber more evenly without creating a large shift in weight across the platform. We maintained flow over the mat at approximately 1,200 gallons per hour, which was calculated based on the 1 meter square area of the mat surface. The scrubber was deployed in the lake on June 14, 2020, and allowed to work for 26 days so that native periphyton could seed the mat. Periphyton and lake water samples were collected 3 times between July and September at 14 to 28 day intervals. We used a battery-powered shop vac to collect periphyton from the mat surface. Water samples were collected from the pump outlet and later analyzed for total phosphorus and heavy metal concentrations. Periphyton samples were dried, weighed, and sent off for nutrient analysis. A sample of periphyton was collected and preserved in Lugal's iodine solution for later species identification. Our average dry mass was 83.79 grams, with an average daily production of 4.55 grams. As expected, the phosphorus concentrations in the North Basin of Wapato are high, with an average of 0.163 milligrams per liter. To put that in perspective, normal levels of phosphorus in lakes not experiencing an influx of excess nutrients range between 0.03 and 0.05 milligrams per liter. Dried periphyton contained an average of 0.31% total phosphorus. Uptake efficiency of total phosphorus by periphyton remained close to a 1-to-1 ratio and showed that as more phosphorus and water became available, the better periphyton was at accumulating it. Based on the dry weight of the collected periphyton, the average uptake of total phosphorus was 9.55 milligrams per day per meter squared. In addition to measuring total phosphorus uptake, we examined the ability of periphyton grown on an algae turf scrubber to filter out heavy metals from the surrounding water. The results indicate that periphyton was very effective in accumulating aluminum, arsenic, cadmium, chromium, copper, iron, nickel, and lead. The bioconcentration factor, or the ratio of heavy metals in the periphyton compared to the concentration in the surrounding water, was very high for aluminum, chromium, and lead by a factor of over 100.
The results from this study indicate that periphyton grown on an algae turf scrubber is very effective at removing phosphorus from Wapato Lake. In addition, it's also effective at removing heavy metal contaminants. The values reported here are similar to those reported in the literature for other large-scale algae turf scrubbers. The recovered periphyton biomass from the August 7th sample was notably lower than the other 2. This may have been caused by a seasonal shift in periphyton species, as the mat community switched from one dominant species to another. Despite this shift, the percent of total phosphorus incorporated into the periphyton tissue over the course of the study steadily increased. It would appear that the periphyton is accumulating phosphorus at or near saturation levels, which are estimated to be 0.35 to 0.42%. This is likely due to the optimal conditions of an algae turf scrubber, which promote greater water-to-cell contact. Finally, the ability of the algae turf scrubber to accumulate heavy metal contaminants was impressive. Most notable were the values for aluminum, chromium, and lead. Arsenic removal was also high, with a bioconcentration factor of 23. This is notable in Tacoma because of the history around the Asarco smelter. The smelter was to blame for the contamination of area water bodies around South Puget Sound with high levels of arsenic, a problem that is yet to be resolved. In conclusion, this data suggests that the use of an algae turf scrubber to remove harmful nutrients as well as heavy metal contaminants from affected water bodies like Wapato is promising. The area needed to remove about half of the incoming phosphorus into the north basin of Wapato would be around 600 meters square. 600 square meters may sound like a large number, but in reality, it only makes up about 6% of the total area of the North Basin. 6% to filter approximately 390 million gallons of incoming overland runoff. Despite the space requirements needed for an algae turf scrubber big enough to filter a large body of water, the benefits of producing a potentially profitable and eco-friendly byproduct must be taken into consideration. The popularity of using algae in biofuels is growing as our need for renewable fuel sources increase. And the introduction of algae into products like slow-release fertilizers can actually help to reduce the introduction of harmful nutrients into our stormwater runoff.
Hi, everyone. We are having some technical difficulties with our last presentation, so our wonderful friends at UW are going to work on that while we get started with some Q&A. We're going to start with a question for Theo. The question is, has there been or are there any plans to sequence Wernickeia to identify if it has toxin-producing genetic strains in this region? And you're muted.
Right. Should be good. Can you hear me now? Yeah. Yes, I have a very nice sample from Robin Matthews up at Bellingham, Western Washington. And that was just submitted for sequencing a couple of weeks ago, so it'll be a little while. That's a Wernickeia sample from, I think, up there somewhere near Bellingham. So we'll see. We'll see what that looks like, yeah.
All right. Thank you, Theo. Our next question is for Sarah. If this technique were ramped up to use as a method for management, how could you dispose of the periphyton with heavy metals?
Good question, and something we were asking ourselves. That would be a whole nother experiment that I'd love to do, is looking at the application of this byproduct in reusable resources. I think ideally incorporating it into some sort of biofuel would be the most profitable. But as far as like a slow release fertilizer, we would have to do some testing to see what could be done with that, especially after the metals data.
Okay. Next, we will have a question for Angelica. Your slide suggests that the shoreline for Coldwater Lake is forested, but perhaps not at Spirit Lake. It seems likely that this could also be a major difference between the two. Have you estimated the end fixation amount from cyanobacteria on the log mat?
I talked a little bit in one of the slides that we did do some initial calculations from our 2018 data, but we found that the cyanobacteria weren't actually abundant enough to be that major missing piece that we were looking for that contributed all that nitrogen. So we still don't actually know the main source of that nitrogen.
And I think I fudged that one up a little bit. I believe those were two different questions. I think one was about the shoreline, one was about the end. So I apologize for that. If I negated anyone's inquiries there, I'm sorry. Okay, let's see. Next question for Sarah. Can you estimate what % of the total volume of heavy metals and toxic algae that is present in the lake was removed by the Summertime Algae Scrubber project? And what would be needed to remove a significant amount?
Actually have not crunched that data yet. So we were primarily focusing on the total phosphorus. The heavy metals portion was kind of a plus and added on towards the end of the project. But that's a very good question and something that would be interesting for us to look at.
All right, and then kind of an add-on to that, would this be feasible to scale up to remove phosphorus at the scale for Wapto or other highly eutrophic lakes?
I believe so. It would be a significant feature, but I believe, especially in the case of Wapto, where you literally have two separate basins, the north and the south, you could almost use one as the filtration basin and then move that filtered water to help keep the south basin, clean, more or less. So yeah, I think that it's totally possible. And then there's finding the use for the collected material afterwards and how that can be best resourced so that it's not just waste.
All right, thank you, Sarah. And it looks like we are kind of at the end of our questions for this session. Uh-oh, it looks like we're having some chatting difficulties, it says. And then we're still trying to get Christian online for his presentation. So maybe if we want to give like a 2-minute break for everyone, hopefully we can get Christian up and in this room because I think we all want to hear his presentation. So we'll be right back in maybe like 90 seconds.
Impeccable timing.
All right, sounds like we're good to go, you guys. Yay! Okay, fantastic. Go, Christian.
All right, thank you so much, first of all, for your patience and the little bit of technical delay there in getting this together. But I'm really glad to be here and be able to present on nanobubbles as a chemical-free method for algae control. So as a sort of brief outline of getting into this presentation, I want to sort of start off by introducing this probably unfamiliar concept of nanobubbles and how these are genuinely different from the bubbles that you and I are traditionally familiar with, and then get into a little bit about the impacts that we see that these have when injected into water bodies, a little bit into the science behind that and what Mueller has been doing as a company to sort of develop this new field of water treatment. And then lastly, get into a couple case studies, talking about where we've installed nanobubble generation or injection technology across the country, and talk about the changes in water quality that we've seen, how long it's taken to see a difference in impact, and the impact on the algae communities, the impact on oxygenation within that water body. So let's start off by talking about nanobubbles themselves and how they're different. Next slide, please. Okay, so as I mentioned, everybody here I'm sure is familiar with bubble in terms of that being just a sort of gas vehicle within a liquid, or in our case within water. And really what it is, gas is less buoyant, or sorry, more buoyant than, less dense than the liquid, and therefore the gas rises to the surface and gets released to the atmosphere. That's what happens with most bubbles that you and I are familiar with. Sort of like a blowing through or air through a straw into water, the bubble rises from the bottom up into the surface and bursts. Now, nanobubbles rather are significantly smaller, even than the smallest bubbles that can be achieved with traditional aeration. And they're about 10 to the third to 10 to the fourth times smaller. So that's 1,000 to 10,000 times smaller in diameter than a traditional microbubble. And what that means is that they really have a significantly higher surface area than traditional bubbles, and that transfers into a very efficient transfer of mass, or the transfer of that oxygen from within the bubble into the surrounding liquid. And what's really unique about these, and we can get into the next slide here, is that nanobubbles, even though they contain gas, their motion is actually driven by something called Brownian motion, and that dominates over buoyancy. So what happens is, rather than rising to the surface and releasing that gas to the atmosphere, the bubble consistently stays in liquid, sort of vibrates in place essentially, and continuously transfers that contained gas into the surrounding liquid. And that's sort of where the comparison between nanobubbles and traditional bubbles stops. Because what's really unique about these, and where they're really distinguished from simply just a gas injection technology, is that the bubble itself has a surface charge. And this isn't really unique to nanobubbles or to bubbles themselves, as all bubbles have a surface charge. But as the bubble gets smaller and smaller, that surface charge gets sort of more concentrated. And when that bubble collapses, when that nanobubble collapses, in itself, that charge is released, and that creates an oxidative impact. And we'll get to the next slide to discuss how the benefits from really efficient oxygen injection and nanobubble oxygenation provides value in water bodies. So what I have separated here is, in the first two rows, how nanobubbles provide value as through just sheer oxidation, really through addressing persistent algae blooms, and then really comparing them against what we normally use for chemical algicides in water bodies. And that's because that oxidation effect, that oxidation impact by the nanobubble, when that bubble collapses, does have a direct impact on sort of the integrity of the algae cell, and then can sort of compromise that ability for the algae cell to survive and thrive and reduce algae blooms. Now, as we know, when we're injecting oxygen into water bodies, we also provide various impacts to the overall health of the water body. And one of the things that's unique about nanobubble oxygenation is that we're able to inject oxygen into water in a form that does not break stratification. So really what we're doing here is we're able to inject oxygen directly into a thermocline, whether that's at the surface or at the hypolymbian, where we're able to provide the most value to that water body. And when we're oxygenating that hypolymbian, we certainly know that in certain anoxic conditions, oxygen can reduce the internal nutrient cycling, reduce metal concentrations, especially methyl mercury concentrations within water, also addresses the heart of odor complaints we see. And then oxygen is also essential to mitigating fish kills that are certainly a negative byproduct of algae blooms. And lastly, due to the unique way that nanobubbles provide oxygen and sort of act as that vehicle for oxygen delivery within the water body and delivering oxygen to the sediment layer, you do see a bit of called sediment or muck remediation. So really turning that sediment from a dark, sort of odorous brown color to, certainly in sandy regions, to a sandier, brighter color. Next slide, please. And this is essentially just a visual representation of what we discovered in the last slide. So in certainly the impacted water bodies that we're treating, we often encounter fish kills, that algae overgrowth, both in planktonic algae and then from suspended algae, that in turn can release algae toxins, especially in harmful quantities during algae blooms. And then in terms of oxygen delivery at the muck layer, we'll routinely see high internal nutrient cycling, especially with phosphorus, and that anoxic condition does create conditions that allow fish flies to thrive. So with nanobubble injection, nanobubble oxygenation, really able to see those water bodies change from that poor, very turbid water quality to increased water clarity, and just create an environment that really does allow fish to thrive. And we'll go briefly to the next slide, just as a visualization for how we are able to provide oxygenation in a method that does not break thermocline, and that really is unique to that neutral buoyancy of the water, of the bubbles. So rather than injecting bubbles at depth and them rising to the surface and breaking thermoclines, really just vertically mixing that water body. In comparison to that, nanobubbles do provide that neutral buoyancy and maintain that thermocline. And I'll very briefly go through the equipment that Mueller manufactures to provide this. We have on the next slide here sort of 3 different main units that we sell or we produce for the surface water and aquatic management space. On the left-hand side, we have the clear model unit, and this is meant to be located outdoors. This is primarily for nanobubble injection at surface conditions, really to impact or mitigate algae blooms, prevent persistent harmful algae, and improve water quality. And then in situations where oxygen is required or in larger installations, then we do have the NEO, which is in the middle, which has onboard oxygen generation. And we can provide this in larger configurations, such as in trailers, as shown on the right. So that's the quick background on sort of Mueller and nanobubbles. And we'll go into the next couple of slides to talk about some of the research that we've done. Mueller has a very active relationship with many American or U.S. universities, where we work with researchers across the U.S. to really understand the fundamentals of nanobubbles and explain why we're seeing the benefits and the unique benefits of improvement in this water body, improvement in overall water body health that sort of goes beyond just the injection of oxygen or what you would normally see with even certainly distinguished from traditional aeration. And I have here, just to sort of put some visuals to it, as a recent installation that we did in the San Francisco area. And this is actually in a brackish water installation, but you can very clearly see the difference between before and after. This is about 8 weeks after installation of the removal of the algae on the surface. But we want to go significantly deeper than this, of course. And on the next slide, provide a brief review of how we impact both algae directly on animals, of providing that impact on algae directly, and then also on the conditions that allow algae to thrive. So across our installations, so far we have observed treatment across various species of planktonic and filamentous algae, certainly on the blue-green algae, Microcystis, Spermopsis, and this is often sort of represented in an increase in water clarity. Certainly the everything treatment from these blue-green soup you can occasionally see, which is associated with that really severe blue-green algae blooms, and then certainly the removal of the surface algae mats as well. But how is this happening, right? That's sort of the ultimate question here. And so that bubble itself, when it is injected into a water body, we find that it has 2 main impacts on the sort of integrity of the algae cell and the ability of that algae cell to thrive. One of which is that bubble does have a surface charge to it, and so it is attached to other charged or polar particles within the water. And in a way, we do see that this is, some of the earlier work has suggested that this may impact an algae cell's ability to regulate or self-regulate its own buoyancy. And then what we believe or we understand currently is the driving impact of that immediate impact on algae cells that we see is the chemical-free oxidation provided by nanobubbles. So that really is on the collapse of that nanobubble. When it does release, we have to say, reactive oxygen species, and that is going to provide sort of compromise that integrity of the algae cell. And then we know with the introduction of oxygen into a water body, we are impacting sort of the root or the heart, impacting the overall water body health. And we're doing this by introducing oxygen, which will certainly reduce nitrogen and phosphorus cycling, and then enabling that water body to sort of promote the growth of healthy microorganisms, beneficial bacteria. And one last thing to note here, some of the early work that we've done also suggests that the oxidation from the nanobubbles does provide not only an impact on the algae cell itself, but also on algae toxins. So that oxidation from that reactive oxygen species is sort of an indiscriminate oxidant, meaning that it will impact not only oxygen cell, but also algae toxins themselves. I want to, just for the sake of time here, sort of breeze. Can we go to slide 11, please? Thank you. So I wanted to get into some of our university research, but I think the most important one to address here is some of the work with Arizona State that we've done, where they've really dived into the oxidation provided by or produced by the nanobubble. And we did this work in conjunction with Paul Westerhoff of nanotechnology engineering and water treatment department. And he really dived down into the creation of that reactive oxygen species on the collapse of the nanobubble. What we find is that this occurs primarily in the environment where we have some sort of external stimulus, such as aeration or mixing, something that's going to cause the collapse of that bubble and release that oxidation. And in that case, that's really where we see the most significant impact on algae cells. And I wanted to highlight this because that is where it's most differentiated between just traditional oxygen injection. Again, for the sake of time, let's go down to slide 14, please. And a bit heavy, I guess, in the beginning here on sort of the background of nanobubbles, what they are and the properties they provide. I want to talk mostly about an installation that we've done recently in the San Francisco and the Bay Area, where we installed 3 of our nanobubble generators. This was in 2 trailers that were provided or supplied, where we installed nanobubbles on about a 10-acre, 120-acre-foot water body, where they're really looking for that combined effect of oxygen injection. They did not want to see thermal or break in thermal stratification. And then also they wanted to see a reduction in algae populations in their lake. This is a public recreational reservoir, and the persistent historical harmful algal blooms did cause routine closures of this water body. This is where they really sought a unique chemical-free solution to not only deep water oxidation, but a removal of the algae in their water body. And we have rather extensive historic information or data on this water, and certainly have documented blooms of Microcystis, Dolichospermum, Lingbia, Benzamenon, and other certainly toxin-releasing algae. So we'll go on to the next slide here. And what we did with this installation, we had 2 nanobubble generators, or 2 trailers, as I mentioned. They were installed on the east side of the lake. Now this, the north side is to the left here, and they are labeled as 1X and 2X. And in here we pumped in 450 gallons a minute of a nanobubble solution that's produced with pure oxygen. And this was injected at depth. And we monitored water quality here with 2 different sondes. We used in situ sondes, which the company donated those to this project. It was very generous of them. And that was to measure water quality, not only at surface, but also at depth in real time. And we looked at parameters such as dissolved oxygen, ORP, phycocyanin levels, and temperature, and several others. And that was really to look at the improvement of oxygenation temperature and the maintenance of stratification in the lake throughout the installation. Next slide, please. And what we saw was quite compelling. So we installed the nanobubble generators. They were officially turned on about August 16th, so 8/16. And we had recorded pretty extensive real-time monitoring. This is from the sensor at depth. Pretty extensive real-time monitoring of the dissolved oxygen conditions prior to our installation. And this was not only coupled with the real-time monitoring that we had done, but also years of historic data that had showed anoxic conditions at depth, about a meter off the sediment layer. And about 2 weeks after the installation of the nanobubble generators, we did begin to see a response in oxygen increasing at depth. And really the metric for us was to get the water body above about 2 ppm. We know that at that level, that's really where it's, the literature suggests you'll provide a stable sort of baseline of oxygen for the water body. And so within about 2 weeks of injection, 2 weeks with the beginning of injection, we did see the the oxygen at depth increased from about 0 all the way up to about 3.5. And I have not updated this with the very most recent data, but we did see that continue to climb up to about a baseline of between 4 and 5, fairly consistently, the water. And this was coupled on the next slide with a very dramatic swing in ORP. So the ORP in this water body, again, along with that, it was on those anoxic conditions. Along with those anoxic conditions, the water was also measured at about negative 400 millivolts of ORP, and this is really just indicative of poor water body health and overall water quality. So these are going to be conditions where phosphorus is very easily released from the sediment, and you're going to have anaerobic microbial activity. And corresponding with that shift in dissolved oxygen, we did see a shift in ORP of about 750 points up to a positive 370, which are conditions where you will see that sequestration of phosphorus, and certainly a shift to nitrification or nitrifying microbial activity. I do have another case study in here, but for the sake of time, I think that I may want to go to slide 21 and just quickly wrap up on the application summary, discussing, again, we've used nanobubbles. We certainly have many installations of nanobubbles for simple algae control installations of filamentous, the control of filamentous and planktonic algae. But a way that it's certainly an area that we're getting into is more hypolimnetic oxygenation as a demonstrated approach to not only control algae, but also address the root cause of overall poor water body health. And just to highlight a couple other sort of points in here is with the reduction of algae, often that comes in combination with reduction in taste and odor compounds, which can be very relevant for sort of drinking water reservoirs. We have, certainly seen installations of, or we do have installations on fish, midge-fly control, and sediment decomposition. So apologize for the technical issues at the very beginning of the presentation. These were a bit unforeseen, but I do really appreciate your time today and certainly appreciate and look forward to any questions. Thank you so much.
Yay, thank you so much, Christian. I'm so glad that we got to hear your presentation. And thank you to all of our presenters. You guys did great. I suppose with this brave new world of online conferencing, there's going to be some bumps that we have to navigate, but I think you all did a great job. So thank you. I think we do have a couple minutes, so maybe, Christian, we can squeeze one question for you, which is, have you seen any impacts to fish from the nanobubbles, such as total dissolved gas, supersaturation impacts?
That is an excellent question, and certainly a question we get often from our fishery customers. With nanobubbles, especially in fishery applications, we would probably tend to use either what we call an enriched nanobubble. It's something nanobubble generated with 40% oxygen, or a high purity nanobubble. And with those higher oxygen content bubbles, fish are very easily able to process the oxygen in those bubbles and prevent things like any gill disease from bubble attachment. It's not an issue. We have no reported cases of fish kills or any negative impacts on fish health.
Great. Thanks so much, everyone. We're going to jump to our breaks. So we have a 10-minute break before our next session, which is our lake ecology session that starts at 2:30. So we will see you then. Thanks, everybody.